Do you know there are types of ELISA? ELISA. There are direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. And we will be talking only today with indirect ELISA. Just like other ELISA, indirect ELISA involves enzyme-linked antibodies, or we call it as labeled antibodies. Indirect here refers to secondary antibodies which bind to the primary antibodies, while primary antibodies bind to the antigens. The process of forming stable covalent bond between the enzyme and the antibody is called bioconjugation. Let's see how is indirect ELISA being conducted. First, antigens are immobilized at the wall of microvalve plates. Next, sample serum is aided and washed. If the sample contains complementary antibodies, then they will bind to the antigens. Next, enzyme-linked secondary antibodies are aided and washed. Lastly, chromogenic substrate, which is the substrate that is able to be converted from colorless to visibly colored by enzymatic reaction, is aided. For the first step, immobilizing antigen, we have to choose antigen for the virus we want to test. The second step is for us to add patient serum. The third and fourth step is to make result detectable and measurable. Indirect ELISA is typically used to determine total antibody concentration. The detection method is via measurement of absorbance. If using only single microtiter, this kind of spectrophotometer is used. As for microvalve plates, this kind of spectrophotometer is used. This is a gift from Group 6. Kindly screenshot if you need this.
ELISA result can be displayed as qualitative or quantitative format. For qualitative, the result determines directly whether the antigen or antibody is absent or present. For positive results, a well that produces visible colors indicates the presence of antigens in the assays, but for negative result, a blank well indicates the absence of antigen or an unrelated control antigen. For quantitative, ELISA data is interpreted to plot a standard curve which is a zero dilution of a known purified antigen. Hence, it is able to determine the concentration of antigen in various samples precisely. How can this assay be applied in veterinary field? Firstly, it can be used to diagnose disease in animals, such as toxoplasmosis. ELISA is able to test both Toxoplasma gondii antibodies and antigen that cause this disease. ELISA can be automated so that a large number of samples can be simultaneously tested. As for equine infection anemia, EIA, the two most commonly used serology tests are the AGID and ELISA, while the ELISA test can detect antibodies earlier than the AGID test. Lastly, feline immunodeficiency virus, FIV. The ELISA is used to detect specific antiviral antibodies circulating in the blood of FIV infection in unvaccinated cat. Other diseases in animals that can also be diagnosed by ELISAs are hookworm, feline infectious peritonitis, feline leukemia virus, and parvovirus. While in research field, ELISA is used to detect potential food allergens in milk, peanuts, and eggs. And this diagram will explain more about the detection process. Not only that, ELISA is able to track the spread of disease during disease outbreak. To test the blood serum for the presence of antibodies against disease-causing pathogens, and this indirectly detect infections. In conclusion, ELISA detection is a two-step process which involves antigen, unlabeled primary antibody, and enzyme conjugated secondary antibody. It has higher sensitivity since more than one labeled antibodies can bind to the primary antibody, and it is more suitable for determining total antibody concentration in samples.